So I will talk about molecular modeling in biomolecular science, and I changed a bit title, adding the word nanotoxicity here, because this is also the area of research which our group mostly involved during the last year. And in some sense, drugs are acting in a positive way on the living organism and the toxic element in negative. So it is common that they have some action, biological action, and there is certain connection between this. And yeah, this is what I am going to talk about. I will start from brief introduction about computer modeling and why do we need it to study biomolecular phenomena, and also in application to drug development or nanotoxicity assessment. Also brief introduction to molecular dynamics, only brief because of course you know all of this method and probably use it. And then the main part of my talk will be a review of the work of our group on simulation of biomolecular system. And here I touch several projects or several tasks which we worked on in previous years and now. So I will talk a bit about computation of free energies, salvation free energies, and log P coefficients. And then I will talk about modeling of biomembranes in general, and particularly about our work on force field parameterization. This is lipid force field. Then also talk a bit about some specific studies of local anesthetics and their action and how they work in lipid membranes, on modeling of amyloid beta peptides membrane, and in the end also talk about our current work on modeling of nanotoxicity. So historically we have two ways of studies of biological objects or living organisms. We have in vivo studies when we studies or make experiment with the organism themselves. Mouses here are very popular in this sense. There are also in vitro studies when we study biological matter but taken out of organism. It can be cells, so it can be even separate parts, separate macromolecules, and studied by different chemical or physical methods. And the development of computer, we also talking about third way of studies of matter. This is in silico study when we investigate the matter by computer modeling and simulations. So what is modeling? Modeling can be thought as experiment on a theoretical model. So in the background we have a theoretical model, which is I can say that it is mathematical description of a real system. For example, it can be, if you study DNA, so it can be model for DNA, given an atomistic model of DNA means that we give atomistic structure of DNA in terms of atomic coordinates. Then we have algorithm, we apply certain set of rules to this theoretical model and get the result. And in computer simulation, we use physical law of nature to evolve the system, and we use supercomputer. This is one of supercomputer PDC, which we are using for our simulations. And in the end, we get results. The results can be compared either with experiment or with some analytical theory. And in fact, we can get all molecular property of the study system, atomistically, the structure, dynamics, and so on. And while we are doing modeling, there are can be many purposes of modeling. It can be just visualization of molecular structure in motion. We see how the molecules look like, how they interact with each other, what's happening when the molecules interact, and produce a nice picture, which also experimental people use often. Modeling can help in interpretation of experimental studies. If you study some system by experimental method, some spectroscopy, we get spectrum, something like this, but often to interpret advanced experiment, we need also some theory and some modeling to interpret what do we see in experiments. Modeling is important just for understanding of molecular mechanism and molecular interactions, because we can see the whole sequence of events, what is the reason or what is consequence in all molecular transformations. And then we see that our simulation produce results which is compatible with experiment. We can even think to replace experiment for, say, extreme conditions or for not yet synthesized molecules or materials, and try to understand whether it is worth to synthesize these molecules would have desirable effect. And this becoming also very wide area of application of modeling. It is used for screening of certain properties in molecular or drug design, including toxicity, because in the simulation it is much easier to create the molecules of a certain structure and test the properties and then decide whether we should proceed with it or not. So computer simulation, in this sense, they reduce guesswork in material or drug design. And also, if we think further, they can also, in principle, it can reduce the need for too much expensive experiment or experiment which can be a matter of concern, like experiments with animals. And then there are different types of modeling, and modeling which I will talk about is called physics-based modeling, that it is based on physical principle. There are also modeling which uses statistical principles, that it is just interconnection, statistical correlation between some input and output, and this leads further to machine learning, artificial intelligence, and so other methods. But we are working with physics-based modeling, which are based on physical laws, and this modeling can be done on different levels. On the most fundamental levels, ab initio levels, we consider system as quantum system interacting by, which is described by the Schrödinger equation. But all this can be used only for relatively small system. When we go to biomolecules, we typically use either classical atomistic approximation, when we have atoms connected by bond in molecules, or even use more coarse-grained models to study larger systems. And the rule of games here is physical laws, that on quantum ab initio level, this is Schrödinger equation, and for classical molecular dynamics, this is Newtonian law of motion. So in fact, in molecular dynamics, we use the same equation which Newton described in 17th century and used them to describe motion of planets, well-known second law of Newton. And now we use this in the same way to compute and predict molecular motion as astronomers in 18th century predicted motion of planet. And that is just picture of hardware of 18th century mechanical calculator. And of course, now we have much better hardware to do such computations. I will say that Newtonian equation of motion, we think that, okay, if we have atomistic molecular level, then we can use classical approximation. We don't need to go to quantum. But the question is how to compute forces between atoms, because this is at some uncertainty. And the courses, again, can be computed either from a quantum mechanical computation, but then we need to do computation at each step of molecular dynamics. It's now it is doable, but still state-of-the-art here is up to, say, 1,000 atoms, and time scale is 150 picosecond. It is certainly not enough to describe by molecular phenomena. So here we need to use approximation, which is force field approximation, that is functions which describe approximately the quantum mechanical energy, which is called force field. And this can do simulation much faster, but exclude description of chemical reactions. So force field data comes as gradients of the potential energy. So formulation of the force field is that we give expression to the potential energy as a function of all atomistic coordinates in the system. And the standard molecular mechanical force field, which is often used in the biomolecular simulation, contains several terms: intermolecular bond stretching and bond bending attraction, torsion interaction along the macromolecular backbone, and intermolecular interactions, which are van der Waals interaction presented as Lennard-Jones potential, and electrostatic interaction due to partial charges of atom indices. And even hydrogen bonds are described by a combination of electrostatic and van der Waals interactions. Information which we get from molecular dynamics, in principle, everything. We can get coordinates of all atoms in each time moment, and such molecular dynamics trajectory takes really large space on disk. It easily can eat up gigabytes and even terabytes of disk space. And from them we can compute, in fact, everything. We can compute various structural properties, radial distribution function, other distribution. We can study coordination of atoms, binding, which ligands bound to which protein. We can compute thermodynamically based parameters and property, such as partitioning, including log P coefficient. We can use this also to describe solubility or crystallization. We can compute thermodynamical properties, enthalpies, free energies. Also dynamical property like diffusion, rate of certain events like permeation of small molecules through membrane, and so on. Okay, so this is introduction, and now I'll come to some application and some investigation which we worked on in our group during the last years. And okay, I begin from log P computations. So what is log P? If we have two non-mixing liquids and we have a third component, then which can be dissolved in both. Then we can measure concentration of these components in both phases, in both liquids, and the ratio of the two concentration, it is the partition coefficient. And because it can be either very large or very small, then often a logarithm of this quantity is used. And this is what is called log P. And the most relevant, or most often computed and measured, is octanol water partition coefficient, that is partitioning between octanol and water. And this is one of the key parameters in drug development, actually introduced for more than 100 years ago by Overton and Meyer. So this parameter, in fact, describes how this lipophilic or hydrophilic is a substance, where it goes to aqueous phase or to lipophilic phase. And this parameter is essentially determine distribution and transport of chemical substances in living organism. This is because octanol can be considered as a small model of lipids which build cell membranes. That we have polar group here and hydrophobic tails, and lipids is just a larger. And for a molecule, for example, a drug, if it's distributed in the organism, you need to penetrate through cell to cell. It means to need to go to cell membrane and then go out if it need go to penetrate inside the cell. And for this, to do this efficiently, log P of the corresponding molecules should be in a certain range. For example, if we have two hydrophilic molecule, it just will not go to membrane and then it cannot penetrate to cell. So this is one of parameters which candidates to drug is screened during drug development. So how to compute log P from modeling? Well, it is from statistical thermodynamics. We know that logarithm of concentrations in both phases is equal to difference in Gibbs solvation Gibbs energies in each of the phases divided by RT. And corresponding log p, which is a decimal logarithm, we need to just put this coefficient 1 divided to about 2.3 to go from natural to decimal logarithm. And the solvation free energy is free energy change due to transfer of a solid from ideal gas phase to the solvent. And this is what can be related in molecular dynamics computer simulation. So typical setup of this simulation is that if you consider molecular in a liquid phase, then we decouple interaction of this molecule with molecules of the liquid phase. And so we describe total interaction energy as a sum of potential energy of the solvent, of the solute, and this is interaction of energy between solvent and solute. And if we put coefficient between this energy to 0, we effectively decouple this molecule from the solvent. And when alpha becoming 0, then we have this case that we have pure solvent and molecular in the gas phase. And the difference in the free energy between these two systems, it is just solvation free energy. That is what we need to compute. And there are different ways to do this. There are many free energy methods, perturbation theory, thermodynamic integration, umbrella sampling. And in our group, we developed and did simulation using the expanded ensemble method. And in this method, this coupling parameter alpha is considered as a parameter of the interaction of Hamiltonian, which is also changed during simulation, and distribution over this parameter give us free energy difference. But I will not go to details because I only overview our research. I will just show our older results. So we did actually this studies for about 20 years ago. It was collaboration with people from AstraZeneca, which at that time they were interested in development of such screening methods. And we did log P computation for a number of drug-related molecules, computed solvation free energy, and from this log P compared with experimental values. And there is some reasonable agreement, but not perfect. You see that for some molecules there is more difference, noticeable difference. But anyway, the trend is followed. More hydrophobic molecules show higher log P values. And here difference with experiment is mostly force field problem. This is not problem of free energy computation, but it is force field problem. And later we did some efforts also to improve force field, just to describe properly free energy of solvation and partition coefficient computation. This was this work with Joachim Jambik. And in this work, we modified GAF, this is general ambient force field parameters, in order to try to better describe solvation free energies. And so we did some modifications, we computed solvation free energy for more than 200 compounds and organic compounds, which also were molecules like this. And this shows mean unsigned error about 2 kilojoule per mole, which produce about half units of mean unsigned error for log P coefficient. So it is somewhat less than we see here. And this size is, in principle, this is acceptable for screening of molecules for desirable values of log P coefficient. And of course, that's now the simulation can be run very fast. So if we did this for 20 years ago, because you need only to have a liquid box, which with this molecular, and such simulation can be run now really fast. So this and it is used really for screening, as I recently heard some talk of representative of AstraZeneca. Okay, so now I'm going to simulation of biomembranes. We also did quite a lot of simulation of biomembranes, and this is very important biomolecular object because cell membrane is composed of lipid bilayer, it's basic construction elements. So lipids are organized in this way with polar groups directed to surrounding liquids and hydrophobic tails inside. Our study was pure lipid bilayers composed of lipids of one type, because this is also this object are studied extensively experimentally to understand properties of lipid bilayer. Of course, real biomembranes are much more complex system that they include many different types of lipids, maybe hundreds of types of lipids in the same in cell membrane. And it includes post-saturated and unsaturated fatty acid chains of different lengths, and combination of different head groups. And also it includes different sterols, of which cholesterol is most important. Also it includes membrane proteins, carbohydrates, and many other things. And of course, what is happening in the cell is crucially depends on what's happening within the cell membrane, because membrane provides transport of everything in and out of the cell. Also it works as protective barrier, and also important that it give place for membrane proteins. Behavior of membrane proteins and also very much depend on the surrounding membrane. So in order to properly describe the system, we worked on development of force field for lipids to describe lipids and use them in simulation of realistic biomembrane, which include mixture of lipids and interactions with peptides or proteins and small molecules. And here we use just standard atomistic description with non-polarizable force field with Lennard-Johnson electrostatic non-bonded interaction. That is what's called standard molecular mechanics force field, which has this form with bond angle torsion interaction, and all these parameters are parameters to fit to provide desirable behavior. So how we parameterize this parameter? The strategy was that what is possible to compute from quantum chemistry, we compute from quantum chemistry. That is partial atom charges. From ab initio computation, it was density functional theory computation, we can compute electron density, and from electron density integrated over atoms, we can compute partial atom charges. Because lipids are flexible molecules, the partial charges depend on lipid conformation. That is why in derivation of lipid force field, we made computation for different lipid conformations taken randomly from already generated molecular dynamics trajectory, computed partial charges for them, and then take average and use these average charges in the final formulation of the lipid force field. Also, torsion angles are very important because the molecular conformation, general molecular conformation, are determined by the torsion angles. We also compute from quantum theory that we use rotation around corresponding torsion angle, computed quantum energy, and fit torsion parameters to reproduce these quantum energies. But Lennard-Johnson parameters were more difficult to fit to quantum theory, mostly because the dispersion interaction of Lennard-Johnson interaction is poorly reproduced by quantum chemical theories, density function theory, or Hartree-Fock theory. One need to go to very advanced quantum theory, but then it takes very long time for computation. So Lennard-Johnson parameters essentially were fitted to reproduce experimental thermodynamic data for small molecules representing corresponding parts of lipids and feeds to density enthalpy data, and also finally to some experimental data for biolayers. And bond interaction we just took from previous charm force field developed previously, and essentially bond and angular interaction are more or less well defined, and there is no much contradiction about them, so that they are easily defined from available structure. And then when we parameterize for available experimental data, comparing properties such as average area per lipid, electron density, X-ray and neutron structure factors, and NMR order parameters. Here's some example of such comparison. So for per lipid, we did computation for a number of lipids for which we find experimental data, and we checked both temperature dependence, so temperature dependent did simulation in some range of temperatures for which we have experimental data. We compare with also NMR order parameters. These data are also available in the literature. And also did comparison with X-ray and neutron structure factors. These are also taken from literature or just by contacting with people. Kusherka did contribute us with data on electron density, and also we get quite good agreement with experimental results. Okay, so development of lipid force field is described in this series of papers. Recent contribution by Frederick Groth, where we improve parameters which describe glycerol and general hair group of lipids. And yeah, now it is parameters are available for saturated and monounsaturated and polyunsaturated fatty acid chains, and also for these lipid head groups, also for cholesterol. So this describes most of lipids which are present in the cells of in the cell membranes of living organisms, and parameters which with ready-to-use files, which we can just directly plug into Gromox, are also available from this site at Zenodo, so they are uploaded there, and so they are in open use. Okay, these are some studies which we carried out with lipid membranes. So one of the tasks which we considered is penetration of drug-related compounds through lipid bilayer. So such penetration or partitioning of small molecules through membrane is best described by the potential of mean force. The potential of mean force can be also understood as free energy of the molecule at certain point in the membrane at certain depths. And this potential of mean force can also describe binding or absorption free energy. In the same way, it allows, as log P coefficient, it allows to compute drug partitioning, that is expected drug density in different points across membrane. Essentially the same formula, that log P is difference in free energy between in water phase and inside membrane. This potential of mean force curves also allows to determine permeation rate of this molecule through membrane due to passive transport. So we studied a range of molecules which are written here: chlorobenzene, chloroform, diclofenac, some others. So we studied both one-dimensional potential of mean force. Here is example. I think the example for lidocaine, but I'm not sure. And also tested, also computed two-dimensional free energy maps, that is free energy on a certain depth of a drug in membrane, and also its orientation in the given depth in membrane. So an example shown here. And for this simulation, we use advanced sampling by metadynamics. But in metadynamics, we use some bias potential, which force a molecule go inside membrane even if potential true potential prevent this. But then this effect of bias potential can be taken back and true free energy profile be reconstructed. So the metadynamics is very efficient method for such computations. So here are some papers when we did this studies on drug elimination. Then in another project, we model local anesthetic molecules and their behavior in lipid membrane. And the local anesthetic, you know that they give effect that you don't use fully lose the feeling in some area of your body into which this local anesthetic was applied. Particularly, it is used in dentistry when they dentist. And there are a range of such molecules which are local anesthetic. You see some examples here, and you see that structure of them is rather different, but the effect is essentially similar, that one loses sensitivity in the place when these molecules are introduced. And so we can ask what is the action mechanism of these anesthetics. And what is known experimentally that anesthetics block sodium conducting ion channels. And if impulse or signal in nerves is transferred by opening and closing of ion conducting channels, which is also voltage regulated, then if we block some of the channels sodium, then the signal cannot propagate along the nerve and we lose sensitivity. But how anesthetic block ion channels, it is not quite well known. It was suggested that some of anesthetic can bind to proteins of ion channel and somehow block them. But you see that structures of such anesthetics are very different. And when you have ligand binding to a protein, typically it's very specific binding and often just changing of little changing of molecular structure, like adenine methyl group, and you do not have binding. But here you see that structures are different. So another point of view is that local anesthetic affect membrane properties, which affect the work of ion channel. So we did simulation of a lipid bilayer, and here we just took model lipid bilayer composed of DMPC lipids and we put their different concentration of lidocaine and articaine, which are two examples of anesthetic used in dentistry. And so we studied different properties, but what was remarkable that when we add anesthetic to membrane, so if you compute electrostatic potential across membrane, then we see that addition of anesthetics increase electrostatic potential inside membrane. And this increase becoming larger and larger concentration of anesthetic. And this difference, this smaller difference is about 70 millivolt, is in principle already enough to change the work of voltage gated ion channels, because working voltage in for voltage gated ion channel, it is just 70 millivolt. So such changes certainly can affect the work of ion channels. And so this can be as a plausible reason that the ion channels are blocked. And results are also described in these two papers. This is some insight why it can be. So we further insight that we also computed contribution from different molecules into charge density and electrostatic potential change in lipid membrane. And it's turned out that changes in the structure and coordination of water and lipids almost compensate each other. But the main increase in the electrostatic potential comes from anesthetic molecules themselves. So this is contribution just from lidocaine and to electrostatic potential, and contribution from what in the PCs here, the black lines, which are smaller. If you look at how anesthetic molecules is typically located in lipid membrane, you see that this is the most preferable orientation, that anesthetic is located in this area just under the lipid haired groups, with this carbonyl group directly to the surface. And this carbonyl group has a dipole moment. And if all the molecules are oriented in this way, this dipole moment gives the main contribution to the increase of electrostatic potential. And if you look at the structure of all these local anesthetics, then we see that we have a similar group in the middle of the molecule. Okay, the next study was behavior of amyloid peptides in neuronal membrane. And it is well known that amyloid beta peptides are related to Alzheimer's disease, that it is supposed that aggregation of this peptide is the main reason of degeneration of brain and failure of brain to work properly. What is also interesting, that if you look at lipid composition of normal brain and brain of a person with Alzheimer's disease, so this data show that in Alzheimer's brain, the content of strongly polyunsaturated fatty acid is reduced very much, especially this acid, which belong to omega-3 class of fatty acids. So its structure is shown here, and it contains a lipid tail with 22 carbon atoms and 6 double bond, that is polyunsaturated fatty acid. On the other hand, it has increased content of unsaturated lipids. So there is certain connection between that the Alzheimer's brain, it contains low content of polyunsaturated fatty acid, and it also contains aggregated amyloid beta peptides. But the connection, it is not clear that can be the reason or consequence, or maybe it is just two facts that occasionally happen. So we did simulations of lipid bilayers composed of fully unsaturated lipids. This is the MPC lipids, of polyunsaturated lipids. This is so-called this DPC lipid. Also, we took bilayers composed of mixture of lipids which resemble composition of a healthy brain and Alzheimer's brain, and also did simulation with cholesterol, because it is also a necessary component of brain tissues. And we add to these membranes AB peptides. Actually, we have not done simulation with full AB alpha amyloid beta peptides because it's too large, but we made simulation with some fragments shown here. And in each simulation, we have 6-9 peptides in each simulation, which were modeled with these bilayers. So some results. So if we look at the first half of amyloid beta peptides in unsaturated lipid, and here is distribution of several residues of these peptides taken on both ends and in the middle of the molecule. So from 0 to minus plus 20, this this referred to membrane, so membrane is located here. So you see that amyloid peptide here is mostly outside membrane, but if you have unsaturated lipid, then the same peptide partially go inside lipid, and so it's concentrated more in the head group area of the lipid bilayer. And if we take the second part of this amyloid peptide, then in the case of unsaturated membrane, it is divided both in the surrounding solvent and partially go inside bilayer. But if we take unsaturated membrane, so this peptide fully goes inside the bilayer. So here you see clear difference in the behavior of this peptide in saturated and unsaturated bilayers. And also we look at the effects of aggregation, that is clustering of this peptide in different cases, and in healthy and in brain with a decomposition. And also we look at in pure bilayers and with presence of cholesterol. So main outcomes from this simulation. So it is clear that polyunsaturated fatty acids cause stronger absorption of amyloid peptides by membranes. Cholesterol, oppositely, it inhibits absorption of amyloid peptides by membrane, but instead it causes accumulation of these peptides at the membrane surface. When amyloid peptides absorbed by membrane, peptides show weaker binding to each other. So they are more clustered when they are on the surface of membrane, but not inside membrane. Inside membrane, clusterization of peptides is weaker, aggregation is weaker. And this may be the reason that it can be easier cleared by amyloid degrading enzyme. However, when amyloid peptides do not like to go inside membrane, but they instead aggregate on the surface, they show stronger binding to each other and form aggregates. So anyway, this simulation showed that lipid membrane compositions, and especially presence of polyunsaturated lipids and cholesterol, it modulated reaction of peptides with membrane and also the aggregation behavior. So this paper describes these results. And the final part of my talk is about our study of nanotoxicity and how molecular simulation can help with this. That is what is the problem here is that nanoparticles are now used in have very wide use in different areas of technology, and they're used even in drug delivery, for example, to deliver drugs. That drug molecules can be put inside in pores of nanoparticles, which taken up by other organisms, and they gradually released too. And it's hopefully drug molecules do good things, that they treat some disease, but what's happened with nanoparticles, also they should not be toxic at least. And nanoparticles can be also occasionally inhaled if they are present in surrounding air or taken up in other ways. And so to nanoparticles, we consider objects which has or particles which has size in at least one of dimensions within the range from 1 to 100 nanometers. And chemical nature of nanoparticles can be very different. It can be carbon-based nanoparticles, carbon nanotubes, graphene, black carbon, other carbon structures. It can be metal oxides. It can be just metals. It can be some organic nanoparticles, polystyrene, nanocellulose, and others. And yeah, as I said, they have very wide range of application, and there is concern how safe are if they are taken by an organism. And what is challenging here is that biological effects of nanoparticles differ from molecular chemicals, because here it is not only chemical composition important, but many other characteristics can be crucially important for biological effects, including toxicity. And this is the size of nanoparticles, its shape, say spherical, ellipsoidal, or fibers, morphology, same chemical composition, but it can be crystallized in different polymorphic form, or it can be different amorphous forms. It can be structural defects, porosity, surface functionalization, and even manufacturing condition. If same kind of nanoparticles but produced from different producers, they may have different properties. And this makes risk evaluation and also regulatory measures difficult, because here we need not only chemical position, but all this, and all this is not really clear how this affects toxicity properties. And several European projects were financed within Horizon 2020 programs to study nanotoxicity, and our group participated in two of them, in smart nanotox and nanosolvent. So some basic idea behind our approaches to evaluate toxicity. So if you are looking at traditional toxicity prediction scheme, that most of them is used on what's called QSAR analysis. This is statistical analysis, qualitative structure, activity relationship, that it use some predictors, it use some adverse outcomes, experimental data for some physical chemical properties of nanoparticles and effects which they have on living organism. And then one use different statistical methods, from simple regression to advanced machine learning and artificial intelligence method, to make prediction based on this analysis. However, it turned out that just for nanotoxicity, it was very difficult to find correlation, because the whole way of from how nanoparticles go inside the organism to the effects, it's very long, and it's difficult to find any correlation except concentration of the nanoparticles. And also, such QSAR method, they act like a black box. Even if they can give the prediction in some cases, there is no information about mechanisms. So another approach to study effects comes from system biology in bioinformatics. In this area, it was introduced such concept like as adverse outcome pathways, that it is how different components of biological system interact with each other, and so that one can track which consequences may have change of whatever in the organism. So such adverse outcome pathways, they are started from some molecular initiating event when nanoparticle meet biological organism. Typically it's happening on their first contact with cell membrane. If it's inhalation, it's alveolar membrane, and it induces changes first on the cellular level, first to the organelles, and so to toxic effect in the organism. Such adverse outcome pathways are developed in system biology. So for many adverse outcome, it is known which effects, which molecular initiation effects lead to certain adverse outcomes. And such molecular initiating events can be some effects, something will happen in a molecular level. It can be sensing of nanoparticle by cell membrane. If it's inhalation, it can be lung membranes, like functionality. Cellular uptake of nanomaterials can cause oxidation of cell membrane. This is example of discussed specific molecular initiating event is as single wall carbon nanotubes is sensed by tool-like receptor protein, which is important protein in the immune system. And clear that all these effects are in reach to model by molecular simulations. So the basic idea of this approach that we can start from nanomaterial properties, and using modeling we can come to in which cases we get certain molecular initiating events which lead to certain adverse outcomes. And further, we can produce descriptors for these properties which leads to just to molecular initiating events. That developing QSAR prediction schemes, we do not go directly from properties to adverse outcome, but we go only this part, predicting molecular initiating event, which is aware of molecular interactions. And how to choose such molecular descriptors which are predictive to molecular initiating event. That if you aim at QSAR type of analysis, then descriptors are numbers, and such parameters of nanoparticle as size distribution, shape, they can be expressed in numbers. But how to characterize material quantitatively? Because we have very different nanoparticles, which can be carbon-based, metal oxides, some advanced ceramics, and so on. It's like compare apples and oranges. So such descriptors need to be relevant. So what was suggested that binding free energy of small biomolecules to the material as can be such predictive descriptors. And why it is so that free energy is a well-defined physical chemical property. It expresses strengths of interaction of biomolecules to the material. And this quantity can be obtained computationally, but it also in principle it's measured experimentally. And also it's directly relevant to phenomena such as protein formation around nanoparticles and membrane permeation of nanoparticles, how lipids, how protein interact with nanoparticles. So that's why we did quite a lot of efforts to compute binding free energy of small biomolecules. And first we considered amino acids, and then we add also lipid components, and we computed binding free energy of such small molecules to a range of nanoparticle or nanosurfaces. And if we say, so these two plots show binding free energy of certain biomolecules, most of them are amino acids, to titanium oxide, rutile, and nanotized surfaces. And we see that such free energy profile can be seen like a fingerprint of how biomatter see nanomaterial. Because this set of molecules, it includes all important chemical groups, hydrophobic, polar, charged, aromatic. So we have this full set of data. We can hope that it can be predictive to predict some toxic effects. And also already we did some test attempt to build QSAR based on a neutral network model, and which was trained by computational data. Computational data were just absorption energies, and also some geometrical characteristic of these nanoparticles, and experimental data were inflammation potential, which is measured as influx of neutrophils in mouse after inhalation of the same set of nanoparticles. And this is some result of prediction. So there is clear correlation that it is possible to build a predictive scheme to predict such toxicity based on result of simulation. And here big error bars, it's essentially explained that in this training set, the number of nanomaterial studies was limited. So there is a hope that as long as the range of studied nanomaterials is increasing, then the prediction of such scheme will be better. And this is what we are doing now in the ongoing nanosolid project. Yes, so some conclusion that so see that molecular simulation can provide detailed real-time atomistically resolved information on molecular structure, motion, and change, and they can be used to give insight how different molecules, and it can be drugs, it can be some important biological molecules, biomarkers, can be toxic molecules, how they interact with cell membrane and other important molecules of the living organism, also to proteins, DNA, and elucidate this way possible mechanism of action. And then molecular simulation can also predict many properties which are important to description and prediction of a certain biological action, which can be used further in drug design or in assessment of toxicity. So these are final slides. This is our current computational chemistry group in the Stockholm University at our department. Also pharma members which are used, which contributed to this research. And these are our computational collaborators in the European project, who did statistical analysis of the computed result and our financing sources. So thank you, and I can ask questions.